For some reason, I put a very long title in the book, but this is the short title, and the long list of contributors and their ever-changing affiliations. So a lot of people here used to be at Macquarie, but have sort of dispersed, and this is now, well, I'm going to be showing work that is, to some extent, the outcome of 2 monthly Skype calls involving 7 countries, 4 continents, and a rather alarming range of time zones. Not alarming for me, I'm in the middle, but so I think these are a couple of themes that have been very strong in this meeting. One is the idea of trait interactions. I could have just put trait correlations there, but there's a lot of work being done on interactions. So, for example, work in which people are looking at how leaf chemistry influences photosynthesis, or the other way around. I won't elaborate on this, but I will, in what I say, give some hints that I think that sometimes the underlying theory is actually quite weak, or the interpretive paradigm, if you like. On the modelling side, there's obviously a strong tendency for us to want to abandon the original idea of plant functional types that served us quite well for a time, but we realise that really grossly fails to represent the diversity of organisms and has leads to all kinds of problems in models. So we're moving towards traits replacing types. A lot of this work, however, has been based on building in actual observed trait or trait environment relationships that might, for example, change. So you can study these in spatial gradients today, but for example, they might change with CO2 if it's just based on the relationships we observe today. So I think a key goal here is the predictability of traits from first principles, whatever they are. Some first principles or other. As Groucho Marx is said to have said, these are my principles, and if you don't like them, I have others. So I've also been looking at the question of how many dimensions, and of course it does depend what you put in the first place. So I'm not looking at plant size here. It's plant size related, so I'm obviously not looking at one of the key, the absolute key traits, because this is all about leaf traits. But the leafy economic spectrum, of course, is extremely well known, and the fundamental relationship here is that specific leaf area decreases with the lifetime of the leaf. But there are additional dimensions, and they appear to be, in our analyses, independent of the leafy economic spectrum. Now, as a modeler, I really like boring situations where there are no correlations between different variables. So I'm really happy to see things being perfectly orthogonal. So when you put in photosynthetic characteristics, so from C13, we estimate the CI/CA ratio, which I like to give its own symbol, chi, because it's so important, it deserves its own symbol. And then obviously photosynthetic capacity. So when you put them in, you discover that they vary sort of independently. This is from some work in a part of a large database that we're putting together across China. So you see there are sampling data points there. There's the Transx, Northeast China Transx, and then there's a lot of work from southwestern province of Yunnan, from central temperate deciduous forests, and then from boreal forests in the north. So a fair range of the climate space of China is sampled, and these are cases where we have sampling essentially the whole flora, or a lot of it, so involving all the different strata. And we have delta C13 and or VC-max and J-max estimated from field measurements. And this is a little bit small, but if you look at top left here, here you see axis 1 and 2, and so axis 1 basically is leaf area, which is independent of everything else, and then axis 2 is the CI/CA ratio, which is also independent, and then axis 3, well, here is axis 3, and what we see there is N area, LDMC, and then going the other way we have specific leaf area, so that's basically the leafy economic spectrum. But then when we actually look at 3 against 4, for example, we discover that photosynthetic traits are nicely orthogonal to the leafy economic spectrum. So all very good, very boring, stuff I like. And we can do this kind of, obviously we can do this kind of empirical analysis. This is very preliminary results at the moment, but using the fourth corner analysis to look at environment trait relationships and showing different patterns, different dependencies for the CI/CA ratio, positive with temperature, negative with VPD, LMA showing the opposite. N area, you see strong relationships, again negative with temperature, positive, sorry, LMA is not the opposite, it's just weaker. It's the opposite for N area, so negative for temperature, positive for VPD naught. So this is from a large sampling across the Australian continent. Wanted to also draw attention to the relatively strong correlation here of the CI/CA ratio with soil pH, which turns out to be a very persistent feature of all of our analyses. Now I'm going to talk about some of these traits, not so much about the leaf economic spectrum. Somebody already mentioned Hendrick's review of the controls of specific leaf area, and that's something we're looking into and trying to quantify in our data, but Wang Han is one of my key collaborators here, is doing a lot of work on that. I'm not going to talk about that, but I will talk briefly about some of the other key traits. Now, leaf size is something I got a bit obsessed by in recent years because I realized the leaf size is supposed to control the delta T, the difference between the leaf temperature and the surrounding air temperature. And I figure this is really, at an elementary level, this is a lot easier than it appears to be in textbooks. So that every biophysics textbook has something about this. But actually, this is the really easy version because this says that the delta T in steady state, which is reached very fast, has to be equal to the difference between the net radiation received at the leaf and the transpiration from the leaf. And then there's the scaling factor here is the products of heat capacity of air and the boundary layer conductance of the leaf, which is well known to decrease with leaf size. And you have to consider the day and the nighttime cases separately, because in the daytime, net radiation is strongly dominated by solar radiation, it's positive. The main control is solar radiation, main component, whereas at night, there isn't any solar radiation and it's always negative. How negative depends on how clear the sky is, but it's always negative. So those are two quite different cases. And then, of course, in the daytime you have transpiration. The nighttime you have little or no transpiration. So in the daytime, you're looking at the balance of two things, net radiation and transpiration. And according to a very simple approach to transpiration, this term, provided there's plenty of water, provided the stomata are open to let it through, so in well water conditions, then you get to a point where the transpiration exceeds the net radiation. And this is widely observed, well known, and it leads to the conclusion that there is a crossover temperature somewhere in the region of 25 to 30, and below that temperature the leaves are warmer than the air, and above that temperature the leaves are cooler than the air. I've tried asking, anecdotal survey of my colleagues, whether leaf temperature can be cooler than air temperature. And some people say, no, this is impossible. The answer I liked best was actually Ian. When I asked him that, he said yes. I said, how do you know? He said, well, I know when I grab a leaf in the middle of the day in Australia, it's cool. So, I should have just briefly mentioned the nighttime case, because in the nighttime net radiation is negative, no transpiration worth speaking of, and so the leaves are always cooler than the air. In case you don't believe it, here's the global pattern. The red is places where the surface, this is from satellite measurements of land surface temperature. It's the difference from the air temperature, which had to be, tediously interpolated to the timing of the satellite overpass. So tedious and excellent work done by a Kawafat. So the red are places, deserts, whatever, where the surface is very hot compared to the surroundings. And if we just look aside from obvious things like, ice sheets, lakes, wetlands, and so on, we can see a pattern that in the wet tropics generally, wet tropics and subtropics, the leaves are the canopy is consistently cooler than the air. That's in July. If you want to see it in January, then obviously you've got nighttime conditions, very low radiation conditions in the north, and looking much hotter in the south. But again, people who like looking at Australia, if you blow up Australia, you see it's red or yellow everywhere, and then it shifts over to blue in just exactly those places where there is a dense forest. And we've analysed this. So So you see here a plot of land surface temperature versus air temperature, and 52 models. One is the Priestley-Taylor model, which is the one that I explained a minute ago. The other is the much more complicated Penman-Monteith model, but they don't give very different results. And basically, well, the one-to-one line is the dashed line here. So you see there's a crossover, which is indeed, actually globally, it's not far above 25 degrees. We can break this down by regions. It's a little bit higher in the tropics and so on, but it's basically a universal phenomenon. Now, I should say, we filter these data very carefully. These are well-watered, dense, intact forests. As soon as you get into the dry regions, as those maps show, things are very different because, of course, these are not many regions of the world. They're not at all well-watered. There isn't that much water, and strata are closing down. And so, over much of the world, the leaves are forced to heat up above the air temperature. But the boundary-layered conductance, and therefore leaf size, is what determines how much they heat up, but also in the wet tropics also determines how much they cool down. So finally, we have a solution to what controls leaf size. In the tropical forests, there isn't any upper limit. Well, obviously there is, but it's not anything to do with energy balance. Non-tropical forests, there is also an upper limit, but it's due to the nighttime heat loss, about which there are very, very few papers written. There are lots of papers about daytime energy balance, but very few about nighttime, but this is a plausible explanation. But in dry climates also there is an upper limit because you've got restricted transpiration and the leaf obviously doesn't want to heat up massively above its surroundings. And so this is from a paper by Ian and many other co-authors, several of them on this list of authors here, showing where the different limitations apply. So you have the green area where there isn't any limitation, daytime limitation in the yellow area, a zone of co-limitation in between, and then nighttime limitation over actually much of the world. Let's move on to the CICA ratio. A specific hypothesis, the least cost hypothesis, was originally set forth by Ian in a paper in American Naturalist a bit over a decade ago, and then more recently, several of us working together on this, we put this into a sort of a mathematical form that explicitly embeds the Farquhar model. I should mention that this differs from the classic Cowan-Farquhar hypothesis and turns out to have greater predictive power. And one way to understand this is to say it doesn't have what I call a metaphysical parameter, lambda. I call it metaphysical because we can't measure it and nobody knows what it is. So this parameter, anyway, in a somewhat less metaphysical way, the optimum value can be determined as a minimization problem. So what we're going to minimize, this is the least cost hypothesis, minimize a carboxylation cost, A being the transpiration cost term, so that relates to the cost of maintaining the transport pathway, which of course involves living tissues, and the B is the cost of maintaining a certain carboxylation capacity, VC max, and we're going to minimize the sum of the unit costs, so the cost per unit of assimilation. It turns out this leads directly to this equation here, which rather remarkably has the same mathematical form as Belinda's version, but it's a different derivation. And some of you may have looked at Roger Dure's poster about this, where he explains why lots of different optimization hypotheses will give you the same formula. So this term here, which Belinda calls G1 because she doesn't like Greek, and I call squiggle because I can't pronounce what it's supposed to be. But the point is that the devil here is in the details because different hypotheses give you different functional forms here for what it actually is. And I'm not going to talk about this. We don't really use that yet. That's sort of hypothetical. But the point is that this relates to the Michaelis-Menten coefficient of Rubisco, which in turn obviously relates to the carboxylation and oxygenation components. So this has some consequences. The optimum value turns out to depend on the relative costs. It's just the relative costs of maintaining these two capacities. It should be independent of external CO2 concentration. Actually, not quite. If you do the exact formula, it should be slightly declining. It should be independent of PAR, but it should increase the temperature due to these various coefficients, and also the eta, the viscosity of water, which is going down with increasing temperature, and that works in the same way. It should decrease. Should decrease with VPD, as is well known, and interestingly, it should also decrease quite strongly with elevation. And cutting a long story short, this again is Will Cornwell's, based on Will Cornwell's compilation of delta C13 data. And for convenience, we do this logic transformation, and we can actually predict quantitatively what these dependencies should be. So these are the predictions based on our theory: 0.054 minus 0.5 minus 0.08. And there are the values that are actually fitted. So those are the partial relationships actually fitted from the data, and they actually agree within their confidence intervals. And as a result, we can predict, for example, in our data set, we can separate out different plant functional types, and you see that they end up in different places. So different plant functional types, according to the data set, they have different typical values. But it was all predicted with one equation. They have different values because they live in different habitats. Well, there are additional controls. I mentioned soil pH. This has been shown experimentally. It's in the data. Soil moisture is a complication when it gets down to sufficiently low values, then stomata do end up more closed, other things being equal, so it's not all a VPD effect. This was from analysis of drying down experiments, and here the story is a bit different because there are big differences between different types of plants. So these are from experiments where experiment is basically simple. You just stop watering the plant. You keep it in a glass house and you stop watering it, and then you keep measuring it every week until it dies. And so there's quite a few of these in the literature. This is showing, sunflower, very rapid decline in this G1 or squiggle parameter with pre-dawn water potential. Examples here from temperate or subtropical trees, which are sort of intermediate. And then this is the cork oak, very tolerant of low soil moisture. And in this case, this data set, Quercus ilex, even apparently going the wrong way, although it's probably more or less flat. So the big differences, and we can see these in flux measurements. So this is analysis by Benny Stocker, who's now based in Barcelona. And among the various GPP data sets, don't have time to describe how he got this, but I just want to draw attention to the fact that there are large differences in the soil moisture response of GPP. FLUE is the light use efficiency, that's calculated taking into account satellite data on the greenness, as a fraction of light use efficiency that's shown under ideal conditions. And you see there are some cases where you get no response, but anyway it never gets dry enough to give you a response. Others where you get hardly any response, all the way down to very near to zero soil moisture. Another cluster where you get about 30% reduction, and then finally some cases where you get about 60% reduction. And we're still looking into what really is the differences between these cases. So moving on to photosynthetic capacity, another optimality hypothesis that actually has a long pedigree. I know that this has been talked about by Graham Farquhar himself and such people for a long time. I just can't find the references in the literature, but somebody can maybe help me with this. The hypothesis is that Vcmax, which is obviously an adjustable parameter in the leaf, it adjusts itself so that the available light is used. No more, no less. And as a result, you get co-limitation by Rubisco and electron transport under typical daytime conditions. That's a conveniently vague phrase, which eventually puts some more meat on it. But anyway, that's the statement that it's been thought about for a long time and commonly observed, but some of its implications are actually quite profound, and I think have been missed until quite recently. One is that Vcmax becomes predictable. It should increase in proportion to light. It should increase weakly with temperature, but not at the rate that Rubisco kinetics increases with temperature. It should be less steeply, and therefore, if you standardize the value to 25 degrees, you get a decline. And here's just some supporting evidence, measurements made on the same plants in times of similar light regime, but very different seasons. Measurements on the same plants in Western Australia. The top is VC max there against temperature. So was measured at the ambient temperature. VC max standardized to 25 degrees, declining. J max doing exactly the same thing, increasing, declining, even respiration doing the same thing. So that's just a seasonal adjustment. And mostly when people measure it, they follow a protocol that says go there in the optimum time of year and make one measurement. You're missing some very important adjustments. And again, of course, we can predict what these slopes should be, and we can fit the values, and we're pretty close. It's a little bit high, but it's on the outer edge of the confidence interval there. But the point is, if it was just determined by Rubisco kinetics, like nearly all models say, it should be much steeper than that. And I wanted to mention also just the recent paper by Scafaro and others where they've actually done sort of thermal acclimation studies on a bunch of species, also shown that the acclimation does involve changes in the amount of Rubisco. So it's not just the activity, but the actual amount of Rubisco changes as part of the acclimation to temperature. Some other correct predictions also follow, including higher VC max in dry environments. That's been observed many, many times. Higher VC max at high elevations, first observed by Kerner and Dima in a very nice paper in the very first ever volume of Functional Ecology. Now we have an explanation for it. And also somewhat lower VC max at elevated CO2. So that's a prediction of what the plant would do under optimization assumptions. VC max goes down. Now this obviously leads us to think about what controls leaf nitrogen, which can be considered as the sum of two components, a structural component and a Rubisco component. We postulate that the structural component should be proportional to LMA, plenty of evidence for that, and the Rubisco component should be proportional to VC max, and VC max should be controlled by the plant. So in other words, in the end, the nitrogen per unit area is controlled by the plant. Universally, in the literature, almost universally, in the literature of both modelling and interpretation of empirical studies, it's stated that the photosynthetic capacity depends on the amount of nitrogen in the leaf. It's ridiculous. Of course it doesn't depend on the amount of nitrogen in the leaf. The amount of nitrogen in the leaf depends on partly on how thick the leaf is and partly on how many proteins there are. And it's just a study recently published in Geosciences where we show the partial residual plots for an area against N structure derived from LMA, N area derived from our estimates of N Rubisco. The blue and the red are nitrogen fixing versus non-nitrogen fixing plants, so there is a slight difference. So we can go further and ask what controls GPP. Now, again, this is something we've known about for a long time. Empirically, GPP is proportional to absorbed par. You can argue that that is why empirical light use efficiency models that are very widely used, for example, to derive GPP from satellite data, you can say that they work because of this, but the point is this is a consequence of the coordination hypothesis. It actually follows that you don't have a saturating response. Over a timescale of weeks or longer, you have a linear response, a proportional response even. So that's why they work. But now we can make a light use efficiency model which has no PFTs in it other than recognizing two photosynthetic pathways. No parameters that are fitted to GPP data. They can be, the two parameters of the model can be estimated independently. Unlike certain other well-known products, there are no discontinuities between biomes based on a lookup table. And finally, a realistic response of light use efficiency to CO2. It's slightly more complex than what I showed before because this is using a more exact solution for the CI/CA ratio. But anyway, the point is basically this is one equation with a few constants. The key point here is that VC max, J max, and CI are now all entirely predicted from the environment. So they've actually disappeared from the equation. You don't need to know them. And the model, it's not perfect, but this monthly GPP from a whole bunch of flux sites, it is at least as good as any other models of this type. Now, this is forced by satellite data, right? This is a light use efficiency model. So we're not simulating everything. But what we are simulating is the light use efficiency. And we can also look at CO2 effects and compare with, for example, the meta-analysis of phase experiments showing what happens to light use efficiency as a result of CO2 and water use efficiency and some other conductants, and they're all in agreement. So I'm just going to end with a slight digression, or speculation, I suppose. Models tend to focus on the community mean or the community aggregate numbers like GPP, and there are obviously good reasons for that, but I think if we're really interested in questions of biodiversity, we want to know, how does this come about? In other words, how does the community as a whole manage to achieve, the optimal GPP and to have the optimal VC max and all this stuff? Now we know that there is enormous trait variation actually within communities. Some of it, we like to think it's linked to spatial heterogeneity, either abiotic or actually caused by the other species, so light gradients, for example, and also temporal variability. But I suggest that the theoretical framework that I've outlined here, although we've only applied it to sort of, basically in the context of predicting means, that we could go further. We could try to model niche complementarity. And I haven't tried to do that, but I can just end with a data analysis from analysis of tropical rainforest trait data in Australia and China combined, showing that when we put in whole plant traits and we put in assignments to dynamic roles, which was done by expert judgment, and therefore might or might not be accurate, but we can divide the species into subcanopy, small pioneer, large pioneer, and climax, and then so climax ones tend to be up this end. So you're seeing the highest VC max and J max because these are up in the canopy, the highest height, and then the subcanopy species tending to have greater wood density, tending to have higher CI/CA ratios, and then again, our friend the leafy economic spectrum being orthogonal to this gradient. Okay, that's all.